Welcome all of you to our session. The chapter we are going to discuss today is from the second year topics. The chapter is human reproduction. Human beings are capable of only sexual type of reproduction, which is involving a number of important events like formation of gametes, mating, during which the male gametes are inserted into the female genital tract, fertilization, development. Then development will be inside the uterus of the mother. We are having in vitro. No, in the uterus we are having in vivo development completely within the body of the mother. You are viviparous, and only after completing the development the baby comes out, that is parturition. So we are having several important events in connection with the reproduction, and most of these activities are occurring only on attaining puberty. The most important event is the first development of gametes, that is called gametogenesis. That is the first event. We are having male and female gametes which are all produced in their reproductive organs called gonads. Testis is the male gonad and ovary is the female gonad. It is in the testis that the male gamete, spermatozoa or sperms are produced. What is the structure of the testis? In mammals, testes are generally outside the abdominal cavity because in the abdomen, temperature is very high, and that high temperature is harmful to the sperms. Sperms are highly specialized, highly sensitive cells which cannot tolerate, which cannot bear the high temperature in the abdomen. So, testes are kept outside the abdomen in a pair of sacs called scrotum. Okay. Scrotum is in the inguinal position, that is, in the posterior part between the two legs, and the scrotum will be having gonads in it that produces sperms only after adolescence. Okay, so only on attaining puberty the testes start producing sperms. Now let us see the structure of the male gonad and the associated structures. The male reproductive system consists of testis, its additional or auxiliary ducts, glands, and the external visible genitalia. So, testes are outside the abdominal cavity within a pouch called scrotum that will be helping in maintaining a low temperature. Our body temperature will be almost 37, and the temperature inside the scrotum will be 2 to 2.5 lesser than that of the abdominal temperature. Okay. Testis is pinkish shaped oval structures, pinkish colored oval shaped structures with 4 to 5 centimeter length and 2 to 3 centimeter wide. Each testis will be covered by a dense covering, and inside you have about 250 compartments which are separated by septa into 250 compartments called testicular lobules, and these testicular lobules are containing 1 to 3 highly called seminiferous tubules. It is within the seminiferous tubules that the sperms are produced. Within the seminiferous tubule, there are two types of cells, namely spermatogonia cells and Sertoli cells. spermatogonia cells are giving rise to the sperms, and Sertoli cells are producing nourishment that will be helping in the development of the sperms. So, outside the seminiferous tubule, you are having interstitial space that are containing blood vessels and some cells called interstitial cells. These interstitial cells are called Leydig cells. So, in a seminiferous tubule, you are having these seminiferous tubules having two types of cells, spermatogonia and Sertoli cells, and in between you have some cells called interstitial cells or Leydig cells. These Leydig cells are producing the testosterone, male hormone, which are the testosterone. Generally, they are called androgens. Okay. And they also produce some immunologically competent chemicals. They are called sus-tentacular cells. They also have sus-tentacular cells. So, Leydig cells and sus-tentacular cells are present in the interstitium, that is in the space between the seminiferous tubule in that matrix. Now, each seminiferous tubule is a highly coiled tubular structure which will be opening to a recte testis. So, the seminiferous tubules open to the recte testis. Recte testis is a reticulum of tubules, and from the recte testis starts a number of small ducts called vasa efferentia. All these vasa efferentia fuse to form an epididymis. Epididymis is a glandular, highly coiled tubular part which will be lying on the top and backside of each testis. This is highly glandular and coiled. It produces nourishment for the maturation of the sperms. This epididymis will be continued as the vas deferens. This vas deferens, this is the epididymis. Okay. This epididymis is making a U-turn and going upward. Okay. It is straight tube, not coiled, and will form what is called the vas deferens. Vas deferens will be passing through the small inguinal canal that connects the scrotum with that of the abdominal cavity, and it will be then expanding to form an ampulla. It arches above the urinary bladder, and after that it forms an ampulla. Okay. There is a pair of seminal vesicles. That is the most important type of axillary gland, seminal vesicle, whose duct is opening into the vas deferens. So, suppose this is the vas deferens, it is the ampulla. Okay. This is the vas deferens that is arching. The duct of seminal vesicle open to that. Okay. This is the seminal vesicle, its duct, and it forms what is called ejaculatory duct. This ejaculatory duct open to the duct coming from the urinary bladder. The duct coming from urinary bladder is called the urethra. So it opens to the urethra. So hereafter, the urine and sperms are passing through a common passage. So urethra is the common passage for both urine and sperms. Seminal vesicles are producing certain secretions which constitute or which forms 70% of seminal plasma. Seminal plasma is the liquid part which is produced by the axillary glands of the male reproductive system. 70% is constituted by this one. It will contain prostaglandins, fructose, some enzymes, etc. Okay. Now, as it passes, it is having a single gland called prostate. This prostate is a single gland, not paired gland. This prostate is on the outside of the urethra. So, urethra passes within the prostate. Prostate is responsible for the remaining 30% of the seminal plasma. Seminal plasma. And as it again comes down, it will be having a pair of small gland called bulbourethral gland, which is also called Cowper's gland. It is also called Cowper's gland. All their secretions are poured into the urethra, and the urethra open at the tip of the penis by the urinary meatus. Opening is called the meatus, urinary or urethral meatus. Okay. So it is opening at the tip of the penis. So what are the axillary glands? Seminal vesicle, which is paired, prostate, which is single, bulbourethral gland or Cowper's. Bulbourethral gland is producing only mucus secretion, slimy secretion that is meant for the lubrication of the penis. Okay, then it opens out. The external genitalia, that is, which can be seen outside. The external genitalia consists of the penis. The tip of the penis is called glans penis, which is covered by a loose fold of skin called foreskin. Foreskin will be covering. The penis is the external genitalia, which is made up of special tissue that helps in erection. It is made up of spongy tissue to facilitate insemination. It will be erected at the time of sexual arousal. And the enlarged part of the penis, called glans penis, is covered by a loose fold of skin called foreskin, which is also called prepuce. It is also called prepuce. Clear? Now coming to the female reproductive system. Female reproductive system consists of a pair of ovaries and its ducts and associated structures. Ovaries are abdominal in position. It is present on the lower abdomen on either side of the vertebral column, connected to the pelvic wall by means of a fold of membrane called mesovarium. It is also connected to the uterus by means of a double folded ligament. So it is held in position. It is just a small oval shaped structure which is located on each side of the lower abdomen, which is about 2 to 4 centimeter length, and it is having germinal epithelium on the surface, on the outer part. You are having an epithelium, and this encloses a matrix. This matrix is called stroma. This stroma has two parts, an outer cortex. It is made up of connective tissues, cortex, and an inner part called medulla. Having epithelium on the surface. This is the structure of a ovary. Oviducts. Oviducts are called fallopian tubes. A pair of them are present, which will be about 10 to 12 centimeter long. That is extending from the periphery of each ovary to the uterus. It is only a small tube. The outer part by which it is open to the abdominal cavity is called infundibulum. Infundibulum is surrounded by some finger-like structures called oral fimbriae, and they are all lined by ciliated cells. By the action of the cilia, the ovum that is released from the ovary is sucked in. Actually, there is no anatomical connection between ovary and the oviduct. Ovum first falls on the abdominal cavity, then they are all sucked to the infundibulum, which is the funnel-like mouth. It leads to the broadest part, namely ampulla. It is the ampulla that fertilization is occurring, and oviduct is continued as a narrow part called isthmus. Isthmus open into the uterus or the womb. Okay, it is open to the womb. The uterus is a single structure which is commonly called as the womb, having a raised part, that is fundus, a body, and a narrow cervical part, which is called the cervix, by which it is connected to the vagina. So, a raised fundus, a middle body, and a narrow cervix. The cavity of the uterus is in continuation with the cavity of the fallopian tube, and here it is opening to the vagina through a narrow canal called cervical canal. Uterus is a thick, muscular, pear-shaped, inverted pear-shaped structure having an outer perimetrium, thick fibrous tissue called perimetrium. The middle layer is made up of very thick and powerful, the most powerful, smooth muscles called myometrium. Myo means muscles. Myometrium. And the inner lining is formed of endometrium, which is glandular, which will be producing thick layers of mucus during the menstrual cycle. Events are occurring in such a way that the endometrium becomes thickened, and that thickened endometrium is a preparation for establishment of pregnancy. Okay, so we are having three parts: perimetrium, myometrium, endometrium. It is only this endometrium that undergoes changes in connection with the menstrual cycle. Myometrium is capable of vigorous contraction and producing sufficient force to cause the baby to come out during parturition. That's about the uterus. Now, coming to the external genitalia. The female external genitalia consists of mons pubis, which is a cushion of fatty tissue covered by skin, and pubic hairs develop here on adolescence. Labia majora are fleshy lobes of tissue that extend from the mons pubis, and it encloses a minor fold of tissue called labia minora, and the opening of the vagina is enclosed by or partially covered by the labia minora. Now, to this, you are having the opening of the urinary bladder. In females, the urinary bladder opened by a separate aperture. Urethral meatus is lying dorsal just below a finger-like structure called clitoris. Clitoris is a structure which is homologous to the penis, that is, the structure is similar to that of the penis in male, and analogous to, that is, it is functionally equal to the glans penis. It is a highly sensitive part. And the opening of the vagina, which is called vaginal opening or vaginal orifice, lies below this urethral meatus. This vaginal orifice is covered by a membrane called hymen. The hymen may be sometimes torn off because of severe exercises, horse riding, cycling, swimming, etc. Or it may be torn during the first coitus. Perhaps it may not be torn off. It may be fibrous and may not be torn off even after repeated coitus. Okay. So presence or absence of this hymen is not an indication of virginity of a female, because it may be torn off because of exercises. It may not be torn off even after coitus. So it is not at all an indication of virginity of a girl. Okay. Now coming to the mammary glands, which are located on the pectoral region of the females. It is physiologically and emotionally connected to the female reproductive structures. A pair of mammary glands are present in the pectoral region. Each mammary gland is having a number of 15 to 20 mammary lobes, which are separate from each other by layers of fat. Okay. Each mammary lobe contains a number of cells called alveoli. These cells of alveoli are secreting milk, which is stored in these cavities of the alveoli. Then from alveoli, the milk is taken into small tubes called mammary tubules. Several mammary tubules join to form a mammary duct, and several mammary ducts join to form a wider ampulla, mammary ampulla. From the ampulla, milk is secreted, or which is coming to the surface of the nipple through lactiferous ducts. So from the lactiferous ducts, milk is sucked out by the baby. So what is the route taken by the milk from the alveoli? Alveolar cavities, then mammary tubules, mammary ducts, mammary ampulla, and then lactiferous ducts. That is the route of the milk in the mammary glands. Then coming to gametogenesis. After studying the structure of the gonads, we are now going to the structure of gametes and how they are all formed. The development and maturation of gametes is called gametogenesis. Though we are having a number of similarities between the gametogenesis of male and female gamete, there are a number of differences. So we have to study them under two separate headings. Formation and maturation of male gamete, that is spermatozoans, is called spermatogenesis. This takes place in male testis only on attaining puberty. Only after a boy has matured into a man, his testes will start producing sperms, and he will continue to produce sperms till his death. Okay, so that is one important difference. We are having spermatogenesis occurring in two stages. In the first stage, spermatids are formed. These spermatids are then undergoing differentiation. They undergo differentiation and produce the highly specialized spermatids, are undergoing differentiation and producing highly specialized spermatozoa. So these are the events. So first is the formation of the spermatid, first stage. But remember that all events are taking place continuously. Only on puberty, you will be having the spermatogonia cells undergoing multiplication. It undergoes multiplication by mitosis and produce large number of primary spermatocytes. Actually, after multiplication, you will be having large number of spermatogonia cells. These spermatogonia cells undergoes again differentiation and give rise to primary spermatocytes, which are all ready for meiosis. Meiosis consists of reduction division. First meiosis, then followed by the second meiosis. In the first meiosis, primary spermatocytes, which are all 2n, that is having 46 chromosomes, undergo reduction division and produces cells which are all only having haploid n. They are called secondary spermatocytes. So primary spermatocytes undergoes first meiosis and produces two equal sized haploid secondary spermatocytes, and the second division follows. We know that second division is an ordinary mitosis similar to that of mitosis. So each secondary spermatocyte undergoes the second meiosis and thus produce a total of 4 haploid cells. So from a primary spermatocyte, 2 secondary spermatocytes and 4 spermatids are produced. So in this production of spermatids, actually we are having a multiplication phase, then a differentiation, that is the growth phase actually. And it is after that differentiation or growth phase, you will be having the primary spermatocytes produced. Primary spermatocytes are the cells that are ready for undergoing meiosis. After first meiosis, you are having the secondary spermatocyte, and after second meiosis, you are having spermatids. 4 spermatids are formed. These 4 spermatids are haploid, round, immotile, ordinary cells. It is having a peculiarity only in the sense that it is haploid. Okay. It is haploid spermatids. This undergoes modifications which are called differentiation. And for this differentiation, it will be remaining attached to the Sertoli cells, which are all the nourishing cells, and undergoes differentiation. So in a seminiferous tubule, you can see all the stages of development. All the early stages are seen towards the periphery of the seminiferous tubule. This is the seminiferous tubule. And advanced stages are towards the lumen of that cavity of that tubule. This is the epithelium that undergoes division and gives rise to spermatogonia cells. You are having spermatogonia cells and Sertoli cells. Sertoli cells. Spermatogonia and Sertoli cells that make up the wall of the seminiferous tubule. Now you are having the primary spermatocyte, then secondary, again spermatid. All these stages can be seen. The spermatids are now remaining attached to the Sertoli cells, and get nourishment till it completes its metamorphic change, that is differentiation, and once the sperms are fully formed, it gets detached. Okay. Now, a fully developed sperm is a highly elongated, motile structure having a head, a middle piece, and a flagellum. Head is spatula shaped or spoon shaped in case of man. The shape of the head is depending upon the shape of the nucleus it contains. It will vary from species to species. In man, it is spoon shaped or spatula shaped or slightly oval in shape. Okay. It contains a nucleus which is haploid, which is slightly elongated and spatula shaped. And into that, you are having a cap-like structure called acrosome. Acrosome is formed from Golgi complex and it produces enzymes called sperm lysins. These sperm lysins are helping in penetrating the membranes around the ovum during fertilization. So it helps in entering into the ovum during fertilization. At the posterior part, there is a pair of centrioles. The centriole that is very close to the posterior part of the nucleus is called proximal centriole. And the other centriole that lies a bit away from it is called distal centriole. So you are having proximal centriole and distal centriole. They are always lying perpendicular to each other, always in the form of T. Arising from the distal centriole, you have axial filaments that are running through the center of the flagellum. Okay. There's that narrow neck between the head and the middle piece. In the middle piece, forming the axis in the center, you are having the axial filaments. And the axial filaments are surrounded by a very large number of mitochondria that are all fusing and spirally coiling it. This mitochondrial sheath or mitochondrial cloud is called nibbon curd. This is called mitochondrial nibbon curd. It provides the energy for the flagellar movement by which the sperm will be moving and reaching the ovum during fertilization. So, mitochondrial nibbon curd. At the posterior end of the middle piece, there is another structure called ring centriole. It is a ring-like structure, but having no similarity with that of centriole, still the name ring centriole to them. And the remaining part is the long flagellum, which is helping them to move. Okay. So, human male will eject about 200 to 300 million sperms during a coitus. Human being can eject about 2.5 to 4 ml of semen. What is semen? Semen contains sperms plus seminal plasma. That fluid is called plasma. Sperm plus seminal plasma. Seminal plasma is produced by the axillary glands. Is called semen. So 2.5 to 4 ml is ejected out, and that will be containing 200 to 300 million. An average, you are having 60 to 80 million sperms per ml of semen. And out of that, for normal fertilization, 60% of the sperm should have normal size and shape. 60% should have normal size and shape. Otherwise, it cannot effect fertilization. And 40% of them, which is having correct size and shape, should have vigorous motility. They should be capable of movement. Then only fertilization will be effective. Okay. Sperms released from the seminiferous ducts will be carried through the axillary ducts and secretions of the epidermis, vas deferens, seminal vesicle and prostate are helping in maturation, in physiological maturation and motility of the sperms. Seminal plasma along with the sperms constitute semen. Okay. Who is maintaining the healthy condition of these glands and ducts? It is the androgens. Androgens which are produced by the Leydig cells are responsible for maintaining that axillary glands and ducts. Is that clear? Now, coming to the formation of female gamete. There are a number of differences. First difference is that a female will produce or start the production of female gametes, ovum, even when she is a fetus inside her mother's womb. Even at the time of development of a female child, when ovary are developed, within that ovary, oogenesis begins. And oogenesis is not a continuous process. It is arrested at certain stages. Second thing, a female will be producing ova only up to her last menstruation day. That is menopause. After that, she cannot produce ova. She produces ova from the first menstruation date to last menstruation time. Menopause is the last, menarche beginning. So it is during this narrow part. Usually a girl becomes mature by the age of 12 or 13, and that is the menarche. And by the age of 50, menopause occurs. So only during this period she is fertile. This is called fertile period. It is only during this fertile period she will be releasing ovum. And ovum release occurring in a cyclical event called menstrual cycle. She produces only one ovum at a time. It is a regular event occurring in a cyclical, regular event that lasts for 28 days. A cyclical event that is lasting for 28 days. That's called menstrual cycle. She produces one ovum in a menstrual cycle. Okay. So, though the early stages of oogenesis start very early, it is not continuous. Okay. So, the germinal epithelium within the, or that is covering the ovary, undergo multiple division, mitotic division during the embryonic development, during which a large number of oogonia cells, gamete mother cells called oogonia cells are produced. A couple of million oogonia cells are formed within each fetal ovary. So, no more oogonia are formed or added after birth. All that oogonia cells are formed even in the fetal stage. These oogonia cells start division and enter into the prophase, one of the first maturation division or meiotic division. It passes the first prophase and then it gets arrested. Each primary oocyte become arrested at that stage. That is called primary oocyte. Now, each primary oocyte gets surrounded by a fold of layer of granulosa cells. So, understand, this produces a number of oogonia cells by multiplication that occurred during the fetal stages. These are all 2N, oogonia cells. Large number of such oogonia cells are produced. These oogonia cells undergo prophase 1 and after that it become arrested. That cell is the primary oocyte which is ready to undergo meiosis. It is in the primary oocyte. Now this primary oocyte will become surrounded by a layer of granulosa cells. This is the granulosa cells. So we are having granulosa cells and this is now remaining in an arrested condition at the primary follicle stage. This stage is called primary follicle. So the primary follicle is that stage in the development where the primary oocyte is covered by a single layer of follicle cells. Okay. This primary follicle will generate, they will degenerate. Primary follicles are formed even at the childhood days. A very large number of them will be absorbed back. They become atreitic. They become absorbed back. Finally, when a girl attains puberty, at the age of 12 or 13, when she attains puberty, there will be only 60,000 to 80,000 primary follicles left in her each ovary. In each ovary, she will be having only 60 to 80% primary follicles in each ovary. All others are absorbed back. These primary follicles now start developing only when she attains puberty. Till that time, it is remaining in an arrested condition. Okay. Now, on attaining puberty, the primary follicle get modified into a secondary follicle. What are the events? In secondary follicle, the number of follicle cells become increased. It becomes multi-layered. And there is a theca developing. You have a theca covering produced. Now, the theca splits into a theca interna and a theca externa. And a cavity develops between the follicle cells. That cavity is called antrum. So now you are having a cavity. This cavity is called antrum. Antrum becomes filled with a fluid called liquor folliculi. Liquor folliculi. So, theca divides into theca interna, theca externa. An antrum develops, become filled with the liquor folliculi. Now, the remaining stages of meiosis 1 occur. Till this time, it was arrested in the prophase 1. It immediately undergoes prophase 1 if it has not completed metaphase, anaphase, telophase, etc. of the first meiosis, and thus it is now eliminating a small polar body. The division of the cytoplasm is highly unequal. It produces a large haploid secondary follicle, secondary oocyte and a haploid first polar body. So the division is highly unequal for cytoplasmic division. Cytoplasm division is highly unequal. Unequal division results in the formation of a haploid secondary oocyte and a very small tiny polar body. So what is the significance of polar body formation? Polar body helps in eliminating one set of chromosomes. That's how it becomes haploid. Okay. Now, it again enter into the second prophase, passes through that second prophase, and enter into a small stage called prometaphase 2, and is then again arrested. So there are two arrests. First arrest takes place in the childhood days when the ovum is in the primary follicle stage. It is actually the primary oocyte. This primary oocyte will be undergoing the first meiosis, undergoes only the prophase 1, thereafter it is arrested. So after completing the prophase 1, it is arrested. This is the longer arrest because it occurs in the childhood days and the remaining stages of development take place only on attaining puberty. Whereas the second arrest occurs at the prometaphase 2. Okay, and it is in the tertiary follicle stage. Tertiary follicle stage, the ovum is now in the tertiary follicle stage. It passes through the prophase 2, and after completing it, it is arrested. So these are the three events that are taking place in the tertiary follicle stage. Now, ovum enter into the last and the final stage of development called Graafian follicle. Fully grown up follicle is called Graafian follicle. During this Graafian follicle development, there develops a membranous, actually that is a gelatinous covering. Gelatinous covering. That gelatinous covering is called zona pellucida. Between the ovum and the surrounding follicle cells, there develops a zona pellucida. With the development of zona pellucida, Graafian follicle is ready to release ovum. Remember, ovum is released even without completing the meiosis 2. Release of the ovum from a Graafian follicle is called ovulation. Release of sperm from spermatids occur from the Sertoli cells will occur only after the spermatids have completed their metamorphosis change. They have completed their differentiation stage. Only after fully development of the sperms will be released. That is called spermiation. Here it is ovulation. Ovulation is under the control of hormones. Remember, all the activities of the reproductive organs are under the control of hormones. The development of follicles, that is under the control of follicle cell stimulating hormone, and the release of the ovum from Graafian follicle, that is called ovulation, is under another hormone called LH, luteinizing hormone. A sudden increase in the LH, that is called LH surge, causes the Graafian follicles to burst and release the ovum. And when an ovum is released, it is only in the secondary ooted stage. It has not completed the second meiosis. It is covered by a gelatinous jelly-like layer called zona pellucida, and around that you have a layer of follicular cells that is called corona radiata. Corona radiata. Now, after the ovulation, what is left out will be developing into a yellow connective tissue mass, which is glandular, which is secretory, which is called corpus luteum. Luteum means yellow. So, yellow body. It is this corpus luteum that produces progesterone. That progesterone will sustain the endometrium, which is in the form of a thickened mass that acts as a bed for the fertilized zygote to come and get implanted, get attached. So corpus luteum will be remaining active for almost 7 days after its formation. If no implantation, no attachment had not taken place by this time, corpus luteum atrophies, disappear. It becomes a white mass called corpus albicans. Corpus albicans. Okay. So, what are the major differences? Remember, early stages of development in a female gamete take place even in the fetal stage of that girl. And after her delivery, after she is born, all multiplication, that is, oogonia cell formation are stopped. No more oogonia is formed or added. Second one, it is not a continuous process. It is having two major breaks. 3, ovulation occur even before the completion of the second meiosis. It has just started second meiosis. It has reached only protometaphase 2, and it is at that stage it is released. Then, in both first and second meiosis, the cytoplasmic division is highly unequal. It produces a very large cell and a very small cell called polar body. So, at the end of the division, you may have only one ovum and 3 polar bodies. So, division is highly unequal, both the first division as well as in the second division. Is that clear? Now, we have studied that in female primates, monkeys, apes, and human beings are belong to the order Primata. There is a cyclical reproductive event occurring called menstrual cycle. The first menstruation begins in puberty, that's called menarche. And in human females, menstrual cycle is repeated at an average interval of about 28 to 29 days. And the cycle of events start from one menstruation till the next one. So that is the cyclical event lasting for 28 to 29 days. That's called menstruation cycle. And during that menstruation cycle, only one ovum is released at the end of, at the middle of the menstrual cycle. There are 3 major stages in a menstrual cycle. All these events are under the control of hormones, which are produced by the pituitary, produced by the ovary. And they will bring about changes both in ovary as well as in uterus. Now, let us see what are the events. In the first maximum 5 days, there will be flow. What is flowing? It is the endometrium that becomes separated, and that separation will be causing cutting of the blood vessels, breaking of blood vessels. So the blood along with the endometrium will be flowing out. That is called menstruation. It is menstrual flow. It may last for 5 days. Now, the pituitary start producing the follicle cell stimulating hormone FSH that act on the follicle cells. So the follicle cells, primary follicle, gradually develop into secondary, then into tertiary, and then Graafian follicle. And it will take almost 13 to 14 days to complete the development. Okay. By this time, follicles are formed. Follicles start producing a hormone called estrogen. It is the follicle cells that produce estrogen. This estrogen is responsible for two important events. One, it will act on the endometrium, so that endometrium, the glandular endometrium, will be undergoing rapid multiplication. Cells become multiplied. That is called proliferation. Thus, it becomes thicker and thicker. It grows in thickness. A cushion-like tissue is formed there. And this estrogen also is essential for the normal development and functioning of female glands and ducts. Okay, so estrogen, once it is maximum produced, when the follicle become mature, that is Graafian follicle, estrogen level will be maximum. When estrogen level is maximum, it will suppress the FSH production by what is called a negative feedback mechanism. Pituitary controlled the development of follicle cells, and follicles produce estrogen, and estrogen level now suppresses FSH production. Now, the second hormone, namely LH, is produced. It will be suddenly released, and a quick increase in the level of LH, that's called LH surge, occur by about the 14th day, and that causes ovulation. So, ovulation is because of the LH surge. This is the event that is occurring in the middle of the menstrual cycle, that is on the 14th day. And the Graafian follicle now develops into a yellow mass called corpus luteum. This corpus luteum starts producing the progesterone. So the progesterone level start increasing slowly after ovulation. By 15th or 16th day, the progesterone level increases. This progesterone will be having two actions. One, it will be helping in maintaining that extra thick layers of endometrium. Progesterone is responsible for maintenance of that endometrium. So endometrium will be remaining thick. That is number one action. Two, it will be suppressing. As long as there is progesterone, it will be suppressing the formation of FSH. So FSH formation is suppressed, so that no more ovum is developing, no more follicles are growing. So progesterone will be suppressing the FSH production, and it will be maintaining the corpus luteum. But corpus maintaining the endometrium, corpus luteum will be remaining active for maximum of 7 days. It started producing by the 15th day, okay, and will be remaining active for a maximum of 7 days, that is up to 22nd day. If no fertilization had occurred, then there is no implantation, and if there is no implantation, corpus luteum atrophies and become regressive. It forms corpus luteum, white mass called corpus luteum. So progesterone level declines after the 22nd, 23rd day. When the progesterone level decreases by about the 25th day, that extra thick endometrium is not sustained. It was the progesterone that sustained the thickened endometrium. That thickened endometrium is no longer sustained because there is declining level of progesterone. So what happens? By about the 25th day, that extra thick endometrium becomes cut off, peeled off, separated from the endometrial wall, and that will be resulting in the bleeding of blood vessels, and allow the blood flow occur by about the 28th or 29th day. So these are the events. Thus, we are having first 5 days maximum, 3 to 5 days is the correct one, where you will be having menstrual flow. Thereafter, from day 6 to day 14, the follicles are growing. So it's called follicular stage. And endometrium is proliferating. So we call it as proliferative phase, follicular phase or proliferative phase. Now, on the 14th day, you are having ovulation. Thereafter, corpus luteum is secreting hormone. So it is called secretory phase or luteal phase, which is the longest from day 15 to 28. Luteal phase or secretory phase, it is longest. Whereas follicular phase is from day 5 to or 6 to day 14 is a shorter one. Thus, you are having FSH and LH playing an important role. These are all the gonadotrophic hormones produced by the anterior pituitary, having its action on the ovum development in the ovary, and that will cause a change in the uterine endometrium also. The overall events are thus completely under the control of hormones. Okay. So this is the menstrual cycle in females. It is the disintegration of the endometrium that is leading to menstruation. So what is the reason for that disintegration? Concentration of the progesterone become lesser and lesser. Okay. And this event of cyclical reproductive activity will be lasting around about for 50 years, up to the age of 50 in a female. And thereafter it ceases. This is called menopause. The menstrual cycle ceases around about the 50 years of age, and that is called menopause. And cyclical menstruation is an indication of normal reproductive phase, and it is happening between menarche and menopause. Now, normally, menstruation will occur once in one lunar month, that is 28 days. It may slightly become extended, it may a bit early also, and it is always regular after some time. Okay, it should be regular. Absence of menstruation may be an indication of pregnancy, but it may also occur because of so many other reasons, because of the poor health condition of the woman, because of shortage of or lesser amount of hemoglobin content and blood content, etc. There may be feeble bleeding. Okay, we very often miss it also. So, absence of menstruation need not always be because of pregnancy, but when pregnancy occurs, there will be no menstruation cycle. Because why it is occurring, we are now going to study. Fertilization and implantation. So males are producing sperms and females are producing ova. And a male will mate, that's called copulation, with a female, and during that time, a male will eject semen into the female genital tract. That female genital tract is vagina. Into that vagina, he will insert his penis and inseminate. Insemination occurs in the vagina. Now, the motile sperms swim rapidly. From the vagina, it reaches the cervical canal, passes through the cervix, reaches the uterus, and finally reach the ambulatory region. How it is moving? It is moving with the help of the flagellar movement. So, with the help of the flagellum, it swims and reaches the ampulla. Ovum released by the ovary should be also present at that time in the ampulla. And it is in this ampulla that fertilization meeting will occur. And for this meeting, that is called fertilization, an ovum should be always there when a sperm is reaching there. And ovum is not always there. It will be available only on a particular period of time in a menstrual cycle. Only after ovulation, a ovum will be released and will be available in the ampulla. That will be remaining live for only 3 to 4 days maximum, within which fertilization should occur. On other times, if insemination occurs, if mating occurs, there is no ovum, that means fertilization is not possible. That is why we say that all coitus need not end up in fertilization and pregnancy. Okay. Fertilization can occur only if the ovum and the sperms are transported simultaneously to the ambulatory region. This is why not all copulation will lead to fertilization and pregnancy. What is fertilization? Fertilization is the process of meeting of these two sexually different gametes. Sperm and ovum should meet together and fuse to form a diploid 2N cell called zygote. That process is called fertilization. During fertilization, a sperm comes in contact with the coverings of the ovum. The acrosome will get dissolved and will produce sperm lysins or acrosins. They are called sperm lysins or acrosins or sperm lysins, which are all digestive enzymes, which will consist of 3 enzymes: a corona penetrating enzyme that helps in penetrating through the corona layers, then a hyaluronidase that will be helping in removing the hyaluronic acid, that is cementing, attaching the surrounding follicles as together. So when that glue is removed, one by one, the corona cells are peeled off, separated off, and you are having zona lysin. Zona lysin will be responsible for dissolution of the jelly-like zona pellucida. Once the sperm touches the zona pellucida, it will bring about certain changes so that the membrane will block entry of other sperms. This is the reason why normally more than one sperm cannot enter into an ovum. Entry of more than one sperm is called polyspermy. So polyspermy is prevented because of the changes that are occurring on the ovum membrane. Once the first sperm comes and touches the zona touches the zona pellucida layer, it induces changes in the membrane that block entry of additional sperms. That it ensures that only one sperm can fertilize an ovum. So secretion of the acrosomes help the sperm enter to the cytoplasm of ovum through these membranes. So membrane penetration is with the help of the secretion of the acrosome. So sperm lysins, they are all called sperm lysins. Okay. Sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. And this induces completion of the meiotic division. Remember, till this time, ovum is in the second prometaphase. It has not completed meiosis. It is arrested for the second time. That arrest is now broken. It immediately passes through the remaining stages of meiosis 2. And a second polar body is again produced. Cytoplasm immediately divides. That division is again unequal. It produces a very small second polar body and a very large otid or ovum, which immediately fuses with the nucleus of the sperm. Remember, during fertilization, only the head part is entering and acrosome dissolved as enzymes, and now the nucleus will be fusing with the nucleus of the female. Okay, and thus a zygote 2n cell is formed, which is called zygote. So the fusion of male and female gametes together will make the cell again diploid, so that the zygote that develops into a baby will be getting hereditary traits equally from both parents, because sperms are bringing one set, 23. The female gamete, ovum, is having again 23. So, equal number of chromosomes. That's how they become paired, 23 paired. And the zygote is thus getting one member of each pair of chromosome from each parent. Okay. Now, we are having 46 chromosomes, 23 pairs. And what is the nature of male and females? In males, we are having 22 pairs of autosomes, that is 44 autosomes. Okay, 22 pairs of autosomes plus an X and a Y. Whereas females are having 44 autosomes plus 2 X's. Okay. So during meiosis, when it is becoming haploid, each sperm will be getting one member of each pair, that is 22 autosomes plus either X or Y. So you will be having two types of male gametes produced. 50% will be having X, 50% will be having Y. Whereas males are all getting 22 autosomes and 1 X. So all X are similar, but males are producing 50% X containing and the remaining 50% Y containing. So what type of male gamete take part in fertilization? If it is the X containing male gamete that take part in fertilization, then the zygote will be having 44A autosomes plus X plus X. So they will be developing as daughters. If it is the Y containing male gamete that take part in fertilization, then the zygote will be having 44A autosomes plus an X and a Y. They are all developing to sons. So sons and daughters are having a chance of being produced in the ratio 1:1, because the males are producing two types of gametes in 1:1 ratio, 50:50. So the chance of producing male and female children are also equal. The probability is equal, 1:1. So who is responsible for the sex of the baby? The type of male gamete. If it is the X containing male gamete, the baby will be a female. If it is Y containing, the baby will be a male. So it is the male parent which is producing two types of gametes, and the type of gamete, male gamete that is taking part in fertilization is determining the sex of the baby. So sex of the baby is determined by the father and not by the mother. Is that point clear? Now, immediately after fertilization, the zygote starts development. The first stage of development is called cleavage. What is cleavage? Repeated mitotic division is called cleavage. But there is one major difference. The cells are not growing in size. So as the cleavage progress, number of cells increase, size of the cells decrease. The daughter cells called blastomeres are not growing. It is by simple mitosis. Okay. Just imagine cutting an apple. The first division is vertical, dividing it into two equal sized blastomeres. So we say that our cleavage is holoblastic, meaning whole cell zygote is dividing and equal. The blastomeres are equal in size. So holoblastic and equal. It produces 2. Second division is also vertical, but at right angle to the first division. Suppose this is the zygote, first division, then second division. So you will be having 4 cells. What is the size of each blastomere now? 1/4. So in first division, you have 2 cells. The size will be half of the size of the ovum. In the second division, you have 4 cells. What is the size of the cells? Blastomeres? 1/4. Then the third division results in the formation of 8 cells. This is horizontal. So first 2 divisions, you just imagine cutting an apple. First one, then like that. We will be cutting. The third is horizontal. Then fourth one will be result in the formation of 16. And from 8th cell stage onwards, it is called morula, a solid ball. Just like a solid ball, it will be going on. So 2, 4, 8, 16 daughter cells called blastomeres. Embryo to 8 to 16 stage is called, these are the stages. Remember, fertilization is occurring in the ampulla and cleavages are occurring as the ovum is gradually traveling through the fallopian tube. It is coming towards the cavity of the uterus. How it is traveling? By the ciliary action. The fallopian tubes are lined by tall columnar ciliated cells. So the action of the cilia are bringing the fertilized zygote to the cavity of the uterus slowly. So as it is passing through the fallopian tube, it is undergoing these events. Okay. So 8 to 16 cell stage is called the morula. Morula, which is a solid ball of cells. Morula. And morula again continues its division and become a hollow ball-like structure called blastocyst. This stage is blastula. It is in the form of a bladder having a cavity inside, so it's called blastocyst. Okay. It further moves to the uterus, and by the time the blastomeres and the blastocyst are arranged into an outer layer called trophoblast, an inner group of cells called inner cell mass. This process whereby the blastocyst become changed into a 2-layered structure is called compaction. It is called compaction. Thus, it becomes differentiated into an outer trophoblast layer and an inner cell mass. It is from the inner cell mass that the embryo is developing. And the trophoblast provide nourishment, okay, and helps in attachment. The trophoblast layer then get attached to the endometrium and inner cell mass get differentiated into the embryo. So after attachment, the uterine cells rapidly divide and cover the blastocyst. As a result, the entire blastocyst is, see, first the blastocyst is covered by the endometrial cells, then it is withdrawn. It is completely covered and thus firmly attached to the uterine wall. This attachment is called implantation. It is this implantation that leads to pregnancy. Implantation will be occurring by about 7th day of fertilization. Implantation will be taking place generally on the 7th day of fertilization. And immediately after implantation, small finger-like projections are arising from the outer trophoblast layer. These are all called chorionic villi. Small finger-like projections called chorionic villi are appearing on the trophoblast cell, which get surrounded by the uterine tissue and maternal blood. And chorionic villi and uterine tissue become very much interdigitated. Suppose these are the chorionic villi. The chorionic villi and the uterine tissue are now becoming close to the, this is the uterine tissue. Thus, they become interdigitated. That is just like this. Finger-like structures and the maternal tissue grows in between. Thus, a close intimacy is established. That is the functional junction between the mother and the fetus. It is called placenta. Placenta will be completing its development by the 12th week of, it will take 10 to 12 weeks. That is in the first trimester. At the end of the first trimester. Trimester means 3 months of gestation it will take. It is this placenta that is acting as the connecting link between the mother and the fetus. And placenta is connected to the baby by means of an umbilical cord through which the fetal blood alone will circulate. So the baby and the placenta are connected together by this umbilical cord, and the fetal circulation comes to this placenta. Okay. This placenta is connected to the embryo through this umbilical cord, which transports substances to and from the embryo. So, placenta is the organic connection that provides materials that are needed for the growth of the baby. Now, it is also an endocrine. It produces human chorionic gonadotropin, HCG. That is the first hormone. Immediately on implantation, it starts producing the HCG. This HCG is tested for its occurrence in the urine to establish, to confirm pregnancy. That test is called Gravida test. So, doctors will ask you to do this test to confirm whether it is pregnancy or not, which is the reason for the absence of menstruation. If it is positive, this means that HCG is present. HCG is produced only by the placenta, and so placenta implantation had occurred. You are pregnant. That is the meaning. It also produces placental lactogen that is responsible for the development of mammary glands and milk, estrogen, progestogens, and later it also causes the release of a hormone called relaxin. Relaxin is also produced by the ovary. Okay. So a hormone called relaxin is also produced, which will be causing the relaxation of the pelvic girdle. The pubic bone are forming pubic symphysis, which are connected by a cartilage that is slightly dilated so that birth canal become wider. Okay. So HCG, HPL and relaxin. These are the hormones that are produced only in a pregnant mother, only during pregnancy. So during pregnancy, other hormones like estrogen, progestogen, cortisol, prolactin, thyroxine, etc. are also increasing. Why? Because the mother has to control the metabolism of her own body as well as the body of the baby. So these hormones will be increasing in level. Increased production of these hormones are essential for the supporting of fetal growth, metabolic changes in the mother, and maintenance of pregnancy. Now, immediately after implantation, from the inner cell mass, there will be some stem cells. Stem cells are cells that continuously grow, multiply, and produce almost all tissues and organs of the body. These inner cell mass are totipotent. meaning they are capable of producing into or developing into any type of tissue, any type of organ. From this inner cell mass, we produce or we will be having all the organs of the baby. Okay, they are all totipotent. And ectoderm, endoderm, mesoderm, these three layers get established. That is why we say that we are triploblastic. And these three germ layers are established during the stage called gastrula. Okay, so these three germ layers give rise to all tissues, organs in the adult. Now, what are the events that are occurring as the baby is growing? At the end of the first month, heart begins to be formed and it begins to pump. So, small, very feeble sound of heart contraction and dilation can be affected, it can be heard. Only an experienced, okay, with the help of that stethoscope, we can hear the sound, the heart sound. At the end of first month, heart is formed and starting functioning. At the second month end, almost all parts of the body are beginning to be formed. The limbs are established. The embryo become elongated and limbs are formed. And by the end of the third month, almost all organ systems are formed, and in the limbs, digits are also formed. So in the first month, embryo's heart is formed. It is the sign of growing fetus, may be noticed by listening to the heart sound. In the second month, fetus develops limbs. Okay, second month, by the end of 12 weeks, that is first trimester, third month, most of the major organ systems are formed. The limbs are also formed. The external genital organs are also formed. So, after scanning, doctors can identify whether all these organs are formed, and even the sex organs can be identified. Now, the first movements of the fetus can be observed on the first movements. Okay, the baby start moving its limbs by the fifth month, first movements of the fetus, and hairs will be appearing on the head during the fifth month. And by the end of the sixth month, that is second trimester, you will be having the eyelids separated, eyelashes are formed, and fine hairs also will be formed all over the body. So, by the sixth month, the baby is opening its eye, eyelids are separating and is looking into the darkness inside the uterus. And by the end of the 9 months of pregnancy, the fetus is fully developed and is ready for delivery. And the average duration of human pregnancy is 9 months. Okay. This is called gestation period. And when a pregnancy is towards the maturation, we call it as the growth is complete, pregnancy is almost complete, it is about to be terminated. Now, the fully developed placenta and the uterus will be causing some signals which will be understood by the brain. These signals called fetal ejection reflex. The signal for parturition originates from the fully developed fetus and the placenta. That is called fetal ejection reflex. This causes mild uterine contractions. These contractions will activate the pituitary to produce oxytocin. This oxytocin is causing smooth muscles to contract vigorously. So, uterine wall begins to contract more powerfully. The more powerful the uterine contracts, more will be the oxytocin produced. So it is a positive feedback. Pituitary produces oxytocin that causes uterine contraction, and uterine contraction causes more hormone to be released. Thus, the power of contraction goes on increasing, and sufficient amount of oxytocin is thus produced. The similarity reflects between the uterine contraction and the oxytocin secretion continues, resulting in stronger and stronger contractions, leading to the expulsion of the baby out of the uterus through the birth canal. Vagina and the cervical canal together is called birth canal. It is through the birth canal the baby is coming from the uterus. So soon after the infant is delivered, the placenta is also coming out. It is called afterbirth. So it will take 15 to 20 minutes for the placenta to come out. Immediately after the baby is out, baby is delivered, we cut the umbilical cord so that baby can be separated from the placenta. Then 15 to 20 minutes after, the placenta is also coming out. And that will be also resulting in the separation of the highly developed endometrium. The highly developed endometrium is also coming out. That will be resulting in severe bleeding. And if for some reason there is no sufficient amount of oxytocin produced, doctors will inject artificial oxytocin, commercial oxytocin. The commercial oxytocin is called pitocin. Pitocin is the commercial oxytocin, and pitocin is commercial ADH. Remember, both ADH and oxytocin are produced by the hypothalamus, stored in the posterior pituitary. So they are conveniently called as pitocin. So doctors will induce delivery by injecting pitocin, and this is pitocin. And mammary glands of the female will undergo differentiation during this pregnancy and start producing milk at the end of pregnancy. That production and release of milk is called lactation. That helps in baby to feed on this mother's milk, and milk produced by the initial few days will be yellowish in color, thick fluid called colostrum, which is very rich in an immunoglobulin IgA, which will be providing very vital antibodies, absolutely essential for their developing baby, for a developing resistance for the newborn babies. And breastfeeding is thus very essential and a must for a newborn, because it is through that the readily digested and absorbed type of food is present in that milk, and it is also giving sufficient amount of immunoglobulins. So breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy baby. So as far as possible, as far as the mother is lactating, the baby should be fed with the milk. Okay, now one more point you have to understand is that endometrium is highly modified as a decidua. That part between the placenta and the uterine wall is called decidua basalis. It is covering the fetus in the form of a decidua capsularis, and the remaining part of the endometrium is lined by decidua parietalis. So you are having decidua basalis, decidua capsularis, decidua parietalis. And there are 4 membranes which are developing from the trophoblast. They are the chorion, allantois, yolk sac, and amnion. The innermost one is the amnion. This amnion produces a watery fluid in which the baby is remaining bathed. That amniotic fluid not only acts as the hydraulic bath that protects the baby from shocks and vibrations, it also protects the baby from dehydration. Yolk sac is very rudimentary in mammals because there is no function for yolk sac because there is no yolk. Yolk is this reserved food material that is present in lower animals like amphibia, reptiles, birds, etc. But here there is no yolk. So it is almost functionless and rudimentary. So we are having 4 membranes and placenta is mainly made up of chorion. So we are having chorionic placenta, chorionic placenta. And the villi are all immersed in the blood of mother. So we call it as hemochorial. Our placenta is said to be hemochorial. So chorionic placenta, hemochorional placenta. These are the 2 terms that you should remember in connection with the placenta. And with that, I come to the conclusion of discussion of this chapter, and we will be meeting with another chapter next time. Until then, take care and thank you. Thank you very much.